Here we go. So folks, what I am doing here is I am trying to assemble a phylogeny, or reconstruct an evolutionary history by looking at the evolution of a single gene that's present in all bacteria, and that's the 16S ribosomal RNA gene. So in bacteria, their ribosomes have several RNA molecules that make up the finished ribosome, and one of them is called 16S, which refers to the size of that particular RNA product. So what I am doing here is you can see there's lots of A's, T's, G's, and C's, which lets us know we're looking at the DNA version of the RNA molecule, or the gene that's found in the chromosome of the bacteria. You can see lots of different names for the different organisms: Salmonella enterica, Clostridium botulinum, Salmonella typhi. I am using a program called Mega, and Mega is nice, even though it's a bit wonky, for a couple of reasons. First and foremost, it is free, which is a very good price. I'm just going to delete that because I've got 2 copies of that. Another thing that's nice about it is it's got its own web browser within it. And this web browser is nice because it has this particular button here, this Add to Alignment button. And the alignment is this colorful diagram that you can see over here. So I am looking for DNA information that has been deposited by researchers around the world, and I'm in this nucleotide search. So I'm going to take you really quickly to the NCBI homepage, National Center for Biotechnology Information. Has many databases that you can search. Today I am looking through. That's one thing about having your own homemade browser, is it's a bit wonky. Come on, go back to where we were. Alright, so we're looking in nucleotide, and we're looking for 16S genes. And I think I am now looking for, I've got Mycobacterium tuberculosis, so the next one I look for is Borrelia burgdorferi. Search, and you can see what comes up. So I was looking specifically for this 16S gene for this particular organism, Borrelia burgdorferi, which is the causal agent of Lyme disease, and you can see what comes up. And we can look briefly over here. We can see here's a related species, Borrelia guerinii, and we've got, it says complete genome. When we look at how much information is there, 904,000 odd base pairs of DNA. That's an entire genome, and we do not want that. We just want a 16S ribosomal RNA gene. This looks a little bit more like it, so I'll click on that. Click. And you can see a lot of information about how this particular organism was cultured, caught, where it was caught from. In this case, it was Korea. If it was used for a journal article, it'll tell you where it was published. And we want to go and we want to find this FASTA file in the form of text. FASTA is just a way of making a file that can be read by a lot of different phylogeny software. So a lot of programs can read this type of file format, starting with this little caret, giving you all this information, and then after this there's full stop. You start with the genetic information. You can see there's all these N's for nucleotides they couldn't resolve, so they just said there were some nucleotides there, we don't know what they were. Anyway, we're going to go ahead and we're going to just click Add that to Alignment. I click Flower 2, and you can see in my alignment that that sequence has been added. Okay, I'm going to embiggen this. big in this, and you can see I've added it to my alignment, which is called an alignment because we're trying to line up homologous nucleotide positions within this gene. You can see there's a lot of areas where the colors match up quite nicely, and some other areas where they don't. So what we're going to do to sort of nicen this up is I'm going to click this W. That W is for Clustall W, which is an alignment algorithm. It's a way of taking nucleotide sequences and trying to match them up so they look nice and neat and match as many homologous nucleotide positions as we can. Nothing is selected for the alignment, so do we want to select all? Yes, that's what I want to do. And you can see here are some parameters. I typically don't screw around with these. I'll just use the defaults. And you can see what Clustall W does is it compares, makes pairwise comparisons between each pair of sequences. And then it takes, after it's done all of that, comparing the pairs, it's going to look at the whole thing overall. And this is kind of a big file because I've got 20 some odd sequences in there. So it'll take a little minute or two, enough time for me to have a sip of coffee. Kick my feet up for a minute. Almost done with the pairwise alignment. Usually multiple alignment will go a little bit more quickly. What do we get? We're about to find out. Alright, so I'll hit flower 2 again. And you can scoot down here and we can see now it all looks a lot more neat and tidy. You can see there's a lot of areas where there's a lot of homology, like this CCC triplet here. Some strains don't have particular regions, so that makes it harder to do the alignment. Some of these really stand out as being very, very different. You can see, like this Clostridium difficile strain, I'm not sure this is the right gene that I'm really interested in because it's so out of left field. That's part of the art of doing sequence alignment, is trying to identify if you're looking really at homologous characteristics or if you're looking at something that shouldn't necessarily be in there. Anyway, I can go back and to my web browser. Whoops, don't want to do that. And this is kind of the tedious part, is typing in my search parameters for each sequence. Again, I've typed in my gene region. Didn't necessarily have to do that first, but it helps me to remember. Now I'm looking for Alii Vibrio. Fisheri. So this is kind of like Google for gene regions. In this particular repository, NCBI, GenBank, let's see, Alii Vibrio Fisheri gene for 16S RNA, partial sequence. It's about 517 base pairs of linear DNA. That sounds about right. We could pick one of these others. I'm going to try this one. Let's see what happens if we do that. Click on that. Here is the GenBank record, which shows you the sequence and all of these other pieces of information collected from India by this person, Neha Thackeray, in India, looking at a bioluminescent bacterial strain. We can go down. We want this FASTA text format. We get the relevant information that we need. Add that to alignment. Okay, and just a couple more, and we'll be ready to go on to the next step. Let's just do one more. Why not? Okay, so here from our search bar, 16S Sphingomonas. Let's go see what's in for. We're not looking for a particular species of Sphingomonas, so we can just search under that, see what comes up. You can see we've got several different species of Sphingomonas in GenBank, and by and large, researchers use the same types of primers, DNA primers, to amplify these genes. So that's why we get all this consistency in this gene region that they've amplified and sequenced. So let's see if they actually have the strain I'm looking for. We're looking for S15-S1. Let's see if that one's in there. Hey, look, there it is. What do we get? We get, yep, this is the one that helps to precipitate out uranium. It can actually process uranium, help do bioremediation. So this is one of the good guys. Let's take a look at its FASTA text. Add that to our alignment. Okay, done that. Go back. Here's another good guy. Let's get the gene region first. Alcanivorax, the alkane eater. Borcumensis. From Borcumen, wherever that is. Aha, here we go. Let's see. Here's one of the strains. Take a look at that. Where was this collected? In Korea. Alright. And we'll go to the FASTA text. And you can see we lose a lot of information about who collected it and where, but we could access that at any time by using the GenBank accession number. So this number is a unique number, which tells us where we could get the rest of the information that we need. But just for brevity's sake, we just add these nucleotides there. So go back, and it's just a couple more. Here's our last one. You can see this is kind of a repetitive process. Searching, and here it takes us right to the isolate I'm looking for. This is another one of our uranium precipitating bacteria. FASTA text. Add that to our alignment. And now I am ready to align everything. So let's see. I want to get everything aligned.